A very warm greeting to all of you. Thank you, Chairperson, for the kind introduction. And I would like to thank FAScinate as well for inviting me to share with you what makes ice cream creamy and smooth. I believe many of us enjoy having ice cream. Have you ever thought what makes ice cream smooth and creamy? This is due to the carrageenan that is extracted from seaweeds. I think when we talk about seaweeds, many of you, perhaps the first thing that comes to your mind is sushi or some other fascinating seaweeds like salad that you come across in restaurants. The fact is, we actually have plenty more of seaweed that you may know of. Seaweeds are also known as microalgae. They are multicellular, photosynthetic organisms that make use of sunlight and nutrients present in seawater, and through photosynthesis process, they produce food. They are categorized into three main groups according to the pigment, which is red seaweed, green seaweeds, and brown seaweeds. Seaweed have many applications. They are widely used for food, medicine, pharmaceutical, and nutraceutical. In the world, there are more than 10,000 species. In Malaysia, we have more than 400 species. Today, I will share with you a group of commercially important red seaweeds that is rich in carrageenan. Euchymatoids are a group of seaweed primarily from genera of Kappa ficus and Euchyma as shown in the picture. They are used for carrageenan extraction. In Malaysia, these seaweed are cultivated at commercial scale in Sabah, mainly in the eastern district, which is Sempruna, Tawau, Kunat, Lahad Datu, and Sandakan. Seaweed cultivation in Malaysia started in the late 1970s on a small scale. Seaweed aquaculture is a very important source of income for coastal community. It is operated by the whole family, as you can see in the picture. The husband and wife are tying the seed before they are out planting in the sea. The picture shows that the seaweed are planting in the sea. On the right picture showing the farmer is planting the seaweed, while on the left, for maintenance during the growing stage, farmer will clean and removing other type of seaweed that might grow on them. The removal of other seaweed is important so that they are not competing with the cultivar for nutrient and sunlight. After culture for the period of 30 to 45 days, the seaweed are ready for harvesting. The farmer are harvesting the seaweed and brought back to the platform for drying. The harvested seaweed are brought to the platform and sun-dried. Once they are dried up to moisture content of approximately 38%, they will be packed and sold to media person or carrageenan factory. These seaweed are primarily used for their carrageenan. However, besides that, they are also popular food among the coastal community. They are either taken as a raw salad or cooked into a dish. The dry seaweed are sold to factory and being processed to extract carrageenan. Carrageenan is a type of linear sulfated polysaccharide. It is used as thickener, stabilizer, or emulsifier in food and drink. These include ice cream and dairy product, jelly, as well as confectionery, and also in cooked processed meat product. Carrageenan is the ingredient that make our ice cream and other food and drink creamy, smooth, juicy, and homogeneous in the texture. Since the establishment of seaweed aquaculture, there is increase of production. However, our seaweed aquaculture facing challenges. After year 2012, we have seen there is a decline of production. There's a reduction of 45% from 2012 to 2020. What are the factors that lead to the decline of the seaweed? One of the factors that leading to this decline is the low genetic diversity among the cultivar. This is because the same cultivar have been propagated vegetatively for more than 50 years. The low genetic diversity may cause the growth rate reduce and potentially lead in size reduction. As shown in the picture, the seaweed size has shrunk. On the left is the size of the seaweed in the mid-70s, and the right picture is the current size. Obviously, our seaweed has shrunk in size. The low genetic diversity is due to the culture technique, which is using vegetative or cutting method. By culturing using cutting method for about 50 years, means that the cultured seaweed are the clone from the same mother plant. With time, this has resulted in low genetic diversity, where the size are shrunk, growth rate reduced, and they are more susceptible to disease. The low genetic diversity was evident in our recent study made in year 2021, where we used molecular marker to see the genetic composition. Based on the two molecular markers, we have shown to have one genotype of Kappa ficus aurelia, two genotypes of Kappa ficus striatus, and one genotype of Malaysianus. This proven that our cultivar is very low in genetic diversity. What is the problem with low genetic diversity? When the cultivar are of the same genotype, that means they have the same immunity or resistance toward pests and disease or climate change. If one of the propagule is susceptible to a certain pest, then all of them will be susceptible. It may lead to risk of whiteout event and may lead to the collapse of our seaweed industry. And this impact can be further intensified with the climate change. Thus, our seaweed aquaculture is in alarming stage, where some mitigation steps need to be carried out. Despite that our country had cultured the seaweed since the late 1970s, but we do not know the species name or scientific name that we are cultivated. Hence, Department of Fishery Malaysia invited me, a seaweed taxonomy, to come in and help to resolve the problem. The seaweed identification is important for trading, as different seaweed will have different type of carcinogen that fetch different price. Most of our euchromoid cultivar are named with local name, with majority are based on how the seaweed look like in appearance. Let me share with you some of the common cultivar with local name. Buaya, having the appearance of rough, uneven scale on the main axis and primary branches, resembling the crocodile skin. Hence, it is named as buaya, the local language that means crocodile. Tambalang giant have three main types. Giant is named as it is due to the large size, generally much larger, denser, and robust. While Tambalang green and Tambalang brown, named after the color appearance. Tambalang is after its own owner name, Mr. Tambalang from Philippines. Sempruna, due to its close proximity to the Philippines. The local names are also influenced by the Philippines. Tangan-tangan is another variety that we are culturing. It has hand-like projection. In local language, tangan-tangan means hands. Green flower and yellow flower is named after the appearance resembling flower as well as the color. Aring-aring is another variety adopted from the Philippines, which is named after Mr. Joey Aring, an owner of a large seaweed farm in Tawi-Tawi, Philippines. Therefore, to address the issue of misidentification and taxonomy confusion, we use DNA to identify the eukymatoid present in Sabah, as well as comparing with eukymatoid in the world. This is because DNA is intrinsic and would not be affected by the external environment. For example, morphological plasticity. Seaweeds are known to be plastic as their appearance will be affected by external growing condition. Our result shows that buaya tangan-tangan tambalang variants are all Kappa Ficus avarezia despite of vast variances in color and appearance. This species is also widely cultivated throughout the world true to its commercial introduction from Philippines. Our result also shows that the flower variants are Kappa Ficus striatus, and the Kappa Ficus striatus are genetically different from the Kappa Ficus found in the wild. Interestingly, the DNA result indicate that aring-aring cultivar is different from Kappa Ficus avarezia and Kappa Ficus striatus, and this could be a new species. Another species is Kappa picosis cottoni was found in Sabah. This seaweed is still untapped and holds great potential in commercial production in the future. Remember, the aring-aring cultivar that was shown in previous slide? Turned out it was a new species after all. We describe this new species as Kappa Ficus malaysianus, with malaysianus derived from Malaysia, which refers to the oceanic region between Southeast Asia where this seaweed could be found. The slide also shows a brief summary of the morphological difference between 6 taxonomically accepted eukymatoid of Kappa ficus and Kappa picosis. To solve the problem of low genetic diversity that we mentioned before, that contributed to low growth rate and susceptible disease, Malaysia need to be able to come up with new cultivar of different quality and resistance. Doing this will add genetic diversity into our pool of eukymatoid cultivar. The best way to achieve this is to look at the wild eukymatoid stock available in the country. The selection and development of novel strain or cultivar is currently the only practical option to overcome the difficulty of cultivar degeneration, disease infestation, or yield loss. Hence, we initiated sampling effort in the coastal area of Semporna with collaboration of Department of Fisheries Sabah in search for natural population with wild eukymatoid. Characterization wild genetic resources is the key for sustaining long-term carcinogen production and may also enable scientists to identify specific threats to wild eukymatoid stock and in turn inform conservation policy. It revealed novel insight into the population genetic structure of this commercial important species with implication for both its efficient conservation and exploitation for breeding. Figure on my left showing the location of sampling site and number of specimens collected in the study. We have sampled from 6 locations, namely Pulau Salakan, Pulau Almarda, Palang-Palang, Pulau Karundingan, Singamata, and Pulau Sebangkat. Also shown are the species that being collected of various eukymatoid indicated by color. Each code indicates a haplotype. In total, we have collected a total of 195 plants where a total of 25 haplotypes are found, and out of that, 17 are novel. Picture shows the morphological variety of Kappa ficus malaysianus and Kappa ficus striatus. On the top are species that was described for the first time in Malaysia, and indeed our coastal water, we are rich in Kappa ficus malaysianus. The result of our study revealed a particular high intraspecific diversity for Kappa ficus malaysianus and Kappa ficus striatus, with multiple novel haplotypes being identified. A high genetic differentiation was also recorded between most population of Kappa ficus malaysianus and Kappa ficus striatus, with Pulau Onwada and Pulau Sebangkat having the greatest number of haplotype. This result suggests the huge potential in exploring the nearby island of Semporna for other novel haplotypes. The wild population are important resources to be developed as new cultivar for seaweed aquaculture and to safeguard this industry. Hence, there is a need to call for conservation. We need to expand the marine protected area to the surrounding island, as this will serve as resources in developing cultivar. By doing so, they will safeguard the aquaculture industry and eventually increase the income for coastal community. By understanding the biodiversity and measurements to conserve the natural population, this will safeguard our seaweed aquaculture industry and for its sustainability. With the increased production, this will ensure a sustainable income for the coastal community. I hope you will join me in this exciting and enlightening journey by protecting our ocean clean and healthy, in preserving the marine organism, in this case is seaweed in particular, so that they may serve as continual resources of cultivar. I would like to end my talk by acknowledging different funding agencies that supported my study. I would like to thank for your attention. Thank you very much.